Hello students, now we are here with our next topic, cytoskeleton. So cytoskeleton basically it is an intracellular network of protein filaments that collectively constitutes supportive structures which provide ultimately support to various cell organelles as well as to the cells, and they are visible under electron microscope. They are not visible under compound light microscopes, and there are three different types of cytoskeletal elements: microtubules, microfilaments, intermediate filaments. And these three different types of filaments, or the components of cytoskeleton systems, they can disassemble or they can assemble to new locations, as well as they are also needed when they are required in the cell to carry out certain specific functions. So assembly and disassembly with respect to these filaments, they are there within the cell, and collectively they provide supportive structure to the cell as well as to various different cellular organelles. In brief, functions of the various types of the cytoskeletal elements, and with respect to their structural as well as their occurrence as well as their functions, where they are found and how they are needed there, they are briefly described here. And you can say the diameter of microtubules, it is around 25 nanometer, while that of microfilaments, the diameter is around 7 nanometer. And in case of intermediate filaments, the diameter, it is on an average 10 nanometer in most of the cases, and it may vary, it may range from 10 to 15 nanometer as a whole. And with respect to composition of these cytoskeletal elements, microtubules are made up of a tubulin heterodimer, that is alpha and beta tubulin heterodimer, and collectively it is called as tubulin protein. This is the monomeric unit of microtubules. On the other hand, microfilaments, they are made up of actin protein. And intermediate filaments, on the other hand, they are made up of different types of proteins. And broadly, these proteins, they are heterogeneous proteins. That may be keratins, they may be lamins, they may be different types of neurofilaments. And the arrangement of these monomeric units in these cytoskeletal elements, it is in such a way that they are polarized one. They form polar filaments in case of microtubules. They also form polar microfilaments with the help of these actin proteins. But on the other hand, intermediate filaments, they are non-polar. It means they the keratins, lamins, or neurofilaments, they are not arranged in a way to form polar skeletal structures, and basically they form non-polar structures. I will explain about the polarity and non-polarity of these cytoskeletal elements in subsequent part of the topic. And in occurrence, all microtubules all eukaryotes, they have microtubules. Similarly, all eukaryotes, they have microfilaments. What intermediate filaments, they are found in certain metazoans, especially in vertebrates, higher vertebrates, and they are absent in plants and fungi. And these cytoskeletal elements, they perform different types of functions. For example, microtubules, they are involved in spindle fiber formation, and subsequently they help in formation of mitotic apparatus, which helps in cell division as well as equal distribution of sister chromatids to the daughter cells. And they also form the structural component of cilia and flagella in case of eukaryotes, and collectively which helps in cell motility. Microfilaments, they helps in formation of contractile ring, and ultimately they helps in division of cytoplasm, that is called as cytokinesis, that is inferred through the formation of a cleavage furrow, where in the cytoplasm side of the cell, actin filaments are arranged in such a way that they form a cleavage furrow. I will explain this components similarly in the subsequent topic. And these microfilaments, especially actin filaments, They also helps in cytoplasmic streaming, and ultimately they helps in cell motility. But on the other hand, intermediate filaments, which are made up of heterogeneous proteins, they have no role in cellular motility, and they have specific roles in organizing cells into tissues. That is, they helps in formation of cell junctions either through desmosomes, or they helps in cell signaling through extracellular matrix as well as to the adjacent cells. As we know, the cytoskeletal elements, they are made up of monomeric proteins which helps in formation of polymers, and these polymers, they are these filaments. And among all cytoskeletal elements or the polymers, they are derived from these monomeric units. These monomeric units, they are held by non-covalent bonds. And coming to the our next first type of cytoskeletal element, it is microtubule. So microtubules, they are hollow cylindrical structures. They are hollow cylindrical structures, and they have approximate diameter of about 25 nanometer. And as I told you earlier, that it is having a hollow cavity, and this cavity, it is also having a diameter around 15 nanometer. Microtubules are made up of a tubulin heterodimer that is called as alpha and beta tubulin subunits. It is a tubulin heterodimer, and during its polymerization these alpha and beta tubulin subunits, they have GTP binding sites and GTP associated with these sites. And during polymerization process, the GTP on beta subunits, on beta not alpha, on beta subunits, it is hydrolyzed, but on the other hand, on the alpha subunit, it is not hydrolyzed, and they are arranged into head to tail fashion on one another, and they form a filamentous structures. You can see the structures here. It is alpha subunit, and it is followed by beta alpha beta heterodimer, and there is also a next subunit alpha beta alpha beta alpha beta subsequently. And during its polymerization, there is unique property here, and with respect to hydrolysis of GTP, the GTP on beta subunit, it is hydrolyzed, but on the other hand, the GTP on alpha subunit, it is not either hydrolyzed or exchanged. So it means this linear structure which is formed or from these tubulin heterodimers, it is called as protofilament, and it has two ends. One end where beta subunits are exposed, and other end there is alpha subunit exposed. And in both the ends, at both the ends, these are protected, or these have GTP associated with these ends, and this form a GTP cap at both the ends. And the ends where beta subunits are exposed, it is called as plus end, and the end of protofilament where alpha subunit is exposed, it is called as minus end. And this polarity is with respect to their N terminal to C terminal in a same linear wave fashion. If I said that C terminal end of protein, alpha protein, it is here, then N terminal, it will be next to this with respect to your beta subunit. There is polarity, means alpha beta alpha beta alpha beta, they form a linear structure that is called as protofilament. And now these protofilaments, they can attain indefinite growth with this polymerization phenomena, and this indefinite growth, it is parallel, or it is as per the requirement of the cells. And subsequently, these protofilaments, they are parallelly aligned to form a ringed structures, ring-like structures, where there is a cylindrical structure is formed, and here 11 to 15 protofilaments, they are parallelly aligned to form this cylindrical structure, and this cylindrical structure, it is called as microtubule. And usually 13 protofilaments, they form a single microtubule. And you can see here also that the alpha subunits of protofilaments, they are found at one end and beta subunits, they are exposed on other end of the microtubule, and this ultimately infer polarity, plus end where your beta subunits are exposed and minus end where alpha subunits are exposed. And this structure microtubule, it is having a hollow center and it is having a diameter around 15 nanometer, while on the other hand the entire protofilament including your hollow cavity, it is having a diameter of around 25 nanometer. And these protofilaments, they are further stabilized here by another protein that is called as MAP. It is a MAP protein, that is microtubule associated proteins, and these maps ultimately bind with these linearly parallelly arranged these protofilaments, and they collectively helps in formation of stable microtubule structures, and they also regulates microtubules with respect to their polymerization and other dynamic properties. As I have told you earlier, that the concept of binding of or association of these tubulin subunits with GTP or GDP, it is an important character for their polymerization and depolymerization phenomena, that is dynamic property of microtubule. And when tubulin molecules carrying GTP, they bind to one another, they have higher affinity to form polymer, and when these GTP, they are lost and they are hydrolyzed or they are exchanged by GDP, then these tubulin, they are not used to form polymers. And presence of GTP cap at both the ends of protofilaments, it encourages a growing microtubule to continue growing, that is polymerization process is switched on. And when the GTP on beta subunit, it is hydrolyzed to GDP, then it is supposed to depolymerize, and disassembly of tubulin subunits initiates from the plus end of microtubules. Further, these microtubules, which are made up of 13 protofilaments that parallelly align to form cylindrical hollow structures, they also can also form doublets, or they can also form triplets. As you can see here, in triplet there is first microtubule that is having 13 protofilaments, and subsequently it is having another microtubule B having protofilament 10, and subsequently third microtubule C having a number of protofilament 10 to 11. These doublet of microtubules and triplet of microtubules, they are found at different sites within the cell. For example, in case of cilia or flagella, microtubule doublets are found in shaft or axonal process of cilia or flagella, while the triplets of microtubules, they are found in the basal body of cilia or flagella. As you can see here, in axonal process of cilia and flagella, they have 9 microtubule doublets, such as this is ring A and B ring of microtubule doublet, first doublet, second, third, fourth, and so on to the ninth. And similarly, they have central sheath that enclose microtubule singlets. There are two single microtubules, and these outer ring of 9 microtubule doublets, they are joined by a protein that is called as nexin. And similarly, these A and B microtubules, they are having dynein arms. These dynein, they are dynein arms, and they are basically motor proteins. And this type of arrangement of microtubule doublets with respect to two single microtubules, it is referred to as 9+2 arrangement, and it is found in shaft of cilia and flagella. And in basal body, they are having these microtubule doublets. For example, A, B, and C. Here you can see the C microtubule of first triplet, it is associated with A microtubule of second triplet, and this form a connective. AC connective, and these collectively helps in formation of a ring-like structures where the central hub is present and it lack any microtubule, and it is different from cilia or flagella, and this type of arrangement it is called as 9+0 arrangement. And they are as I have told you earlier that these microtubule triplets are found in basal body, and they are also found in centrioles, in the centrioles of cells. So next topic, how the origin or nucleation or polymerization process of microtubule is initiated within the cell cytoplasm. So it is a MTOC term, that is microtubule organizing center or microtubule organizing complex, that is located within the cell, and it is located in the cytoplasm near nucleus. And this MTOC is basically rich in a type of tubulin protein, and this tubulin protein is called as gamma tubulin. And in case of animals, MTOC is also associated with a pair of centrioles, and this pair of centriole along with a pericentriolar material rich in gamma tubulin collectively form a structure that is called as centrosome. So here in the diagram you can see that there is a perpendicular position of pair of centrioles with respect to one another, and they are surrounded by a pericentriolar material, and this pericentriolar material on its peripheral site it is having gamma tubulin proteins, and they are highly concentrated in its peripheral region of your pericentriolar material. And this collective arrangement of centriole as well as pericentriolar material rich in that is rich in gamma tubulins, it is called as centrosome, and they are found both in animals and plants, but in case of plants they lack centrioles, and these centrioles basically they are absent in higher plants. And animals they have centrioles, and these centrioles they have specific role, they have specific purpose, as they cause orientation of mitotic apparatus during subsequent cleavage mitotic cell divisions. During embryogenesis they have important role in organizing the cells to different positions. So multicellularity it is attained with different orientations and with different appendages or forelimbs and hind limbs along with different body axes they are formed. On the other hand, in case of plants, they have limited orientation of mitotic apparatus. And here the common link between plants and animals with respect to centrosome is that they have gamma tubulin subunits. And these gamma tubulin subunits they play an important role in nucleation of microtubules, that is process of formation of polymers of microtubules from alpha beta tubulin heterodimers. So from the diagram you can see that the peripheral region of centrosome it is rich in gamma tubulin subunits. Here these gamma tubulin subunits they are associated with these pericentriolar material. This is pericentriolar material, this is pericentriolar material or pericentriolar material here, and these are associated with gamma tubulins, and these gamma tubulins they basically act as foundation, that is base or substrate for your alpha beta heterodimer of tubulin to bind with this and then subsequently process of polymerization. The binding of first few alpha beta heterodimer to the gamma tubulin it is called as nucleation process, and where these gamma tubulins they are located this form a slow growing minus end, and here alpha subunits they are exposed of these microtubules, and similarly they add on these tubulin dimers and subsequently protofilaments and these parallelly aligned protofilaments in microtubules, they are formed having plus end, fast growing end, and minus end, that is slow growing end. And ultimately these microtubules, they are also called as spindle fibers during the cell division. So microtubule dynamics, next topic. It is important topic from a competitive exam point of view, microtubule dynamics. So here we will learn about different features or properties of microtubules related to its polymerization, depolymerization phenomena. So first property is polymerization. It is a process whereby nucleation elongation mechanisms are studied or they are carried out. And here first is formation of microtubule nucleus, and second, this microtubule nucleus, it is further prolonged, and this is called as growth of microtubule, and it occur at both ends by reversible non-covalent. As I have told you earlier, the addition of monomeric units, whether it's your microtubule, microfilament or intermediate filaments, the addition of monomeric unit to the polymer, it is always and always non-covalent one. And these reversible non-covalent tubulin subunits, they are added during elongation phase. And this polymerization, it is further categorized into three phases. Lag phase, which corresponds to the time taken for nucleation process, where your first tubulin heterodimer of alpha beta subunits, they form link with gamma tubulin of centrosome. And second phase is log or growth phase. Here, tubulin subunits are added to exposed ends of growing filaments, that is the plus end of microtubule, and here elongation is achieved. And here beta subunits, they are exposed one. And third phase is steady phase, and it is a unique phase during polymerization phenomena of microtubules, because here rate of polymerization is equal or it is balanced by depolymerization phenomena. And here concentration of free tubulin subunits at plus end or minus end determines rate of polymerization, that is how much of tubulin heterodimers they are added per second. And here concentration of free tubulin subunits, it play a critical role, and the available concentration of these free subunits, tubulin subunits, at this point, it is called as critical concentration. So it is important point, because this critical concentration, it is concerned with steady phase, where the rate of polymerization is balanced by depolymerization, and you have just remaining free tubulin subunits at its plus or minus site of growing, or you can say the microtubules, so formed microtubule. And these available concentration, where polymerization and depolymerization are balanced one, they form a concentration that is called as critical concentration. And these critical concentrations, they are also concerned with another property that is called as tree admilling, because as I have told you earlier, plus end they are fast growing and minus end they are slow growing end. And here this rate is, it is determined by free tubulins at both ends during steady phase of the polymerization phenomena. And this lead to overall chain length that should remain same or unchanged. But what is tree admilling with respect to this critical concentration? So as your plus ends, they are fast growing, and minus end, they are slow growing end. Okay, so if something happen to cell, that the concentration of available free tubulin subunits, they are more at plus towards its plus side, and they are less towards its minus end. So here growth can occur at both plus or minus end site, but the fast growing end or slow growing end, they can be compensated, as it should bring the concentration of these free available tubulins at both ends somewhat towards equal concentration. So when you have a low free tubulin concentration at towards its minus end, then there will occur depolymerization So it will allow the release of tubulin subunits. And plus end, where free tubulin subunits, they are at high concentration, they are added. Suppose I have colored these with blue color and they are added subsequently from this side, and these red colored already they were present, these tubulins were present within the cell, they go on depolymerizing, and this bring about the concentration of these free available tubulin subunits same at both ends of the microtubule. Next property is dynamic instability. So basically it is a phase transition or switching between phases of slow growing and rapid shortening at ends of microtubule, and it is achieved due to hydrolysis of GTP to GDP plus inorganic phosphate, which is bound to plus end beta tubulins after it has been assembled to this process. And this hydrolysis of GTP at plus end or beta tubulin, exposed beta tubulin, it triggered depolymerization phenomena, and this lead to microtubule shrinkage, and subsequently this shrinkage phenomena, it is called as catastrophe. And this catastrophe, it can be controlled or it can be compensated by addition of a GTP bound beta tubulin, which form a new cap and it protect from shrinkage, and this phenomena, it is called as rescue. And this property of catastrophe and rescue, it is collectively called as dynamic instability, which is basically a phase transition or switching between phases of slow growing and rapid shortening at the ends of microtubules. And next, there are certain other proteins, microtubule associated proteins, and they are collectively called as motor proteins, such as kinesin and dyneins. I will explain these proteins in detail in subsequent lectures, but in brief, kinesin proteins, they are plus and directed proteins. It means they carry cellular cargo from one side to the other side, and these orientation or movement of these vesicles, they are determined by the polarity of microtubules as well as the movement of associated motor proteins. For example, kinesin, they move towards plus end, and dyneins, they move towards minus end. This is this is motor protein here, this is motor protein, it is dynein, and it is here kinesin. So kinesin, it is plus end directed, and dyneins, they are minus end directed. These dynein proteins, they are also found in axonal, they are in shaft of cilia and flagella, and they are involved in formation of or the growth of microtubules at the tip or the tip portion of flagella or cilia. And next, these are the drugs. Nowadays, in number of competitive exams, there are number of questions which are directly placed from the topic uh, interfering agents with respect to polymerization and depolymerization of subunits of the microtubules, whether it's microfilaments. And there are certain drugs or chemicals such as nocodazole. They cause basically, they cause microtubules to depolymerize. They they promote depolymerization of microtubules. Colchicines, basically it is a chemical that is used in number of lab experiments also, and it binds with tubulin subunits and it prevent their polymerization. First one, they promote depolymerization, and second one, they prevent. First one, they promote, second one, they prevent. And Taxol, which is also derived from Taxus baccata, and it binds to microtubules and basically it prevent depolymerization. First one, depolymerization promoter, colchicine, it prevent polymerization, Taxol prevent depolymerization, and there are other vinblastine, vincristine. They also prevent depolymerization. So this is all about microtubules. I have told you about their structure, about their dynamic properties, polymerization, triad mailing, dynamic instability. There are motor proteins, and there are number of drugs which influences the working and assembly or disassembly of microtubules during different phases of the cell cycle. If you liked my video, you can share it, comment on it, and can subscribe my YouTube channel for many more such videos. Thank you.